My name is Dr. Schuliger and welcome to my lab. Yes, this is what my face looks like from the bottom half. Living organisms like you and I go through our life, the events in between birthing and dying, or womb to tomb as people call it. Though, have you ever wondered about the life of the smallest building block of all living things look like? Today we will be investigating the microscopic life of a cell. This is the cell cycle. So before we begin to investigate the life of a cell as a whole, let's take a peek into my microscope to understand what codes each cell. Whoa, check it out! It's a DNA strand full of nucleotides that code genetic information that make who we are. The DNA wraps around proteins called histones to create nucleosomes. From there, the nucleosomes tangle up into chromatin, which is currently in an uncondensed form. Let's push it all together. There we go. Chromatin winds around and creates chromosomes. This is the genetic package for all cells, located inside the nucleus or nucleoid. When the chromosomes duplicate, they are still considered one chromosome, just composed of two identical sister chromatids.
Hi sisters, hi sisters.
Where chromatids are connected most closely is called a centromere, kinda looking like a waist. Snatched! On either side of the centromere, there is a kinetochore. It sounds like connect, and the kinetochores connect to microtubules. Kinetochores, connect, easy peasy to remember. There are a certain amount of chromosomes each organism maintains in their cells. Humans have 46, elephants have 56, and the plant the Adler's tongue has 1,260. There are two kinds of cells that make up organisms. The somatic cells are body cells that maintain all the chromosomes each organism has. They are considered diploid cells, with two sets of chromosomes from each parent. Thanks, guys. Gametes are reproductive cells that have half of the normal chromosome numbers. They are haploid cells with only one set of chromosomes. In order for somatic cells to divide, they go through mitosis, the act of replicating chromosomes and dividing the cell to create two somatic cells, both with a full set of chromosomes. In order for gamete cells to divide, they go through meiosis, where after dividing creates four cells, each with half of the needed chromosomes. Here, for example, a human has 46 chromosomes. Somatic cells, like skin cells, have 46 chromosomes as well. And gamete cells, like sperm cells, have 23 chromosomes, exactly one half of the chromosomes somatic cells have. The cell cycle is the life of a cell, from being a daughter cell itself to splitting into two more identical daughter cells. The cell cycle is in a circular shape, hence cycle, and it is divided into stages of interphase and mphase. Interphase makes up 90% of the cell cycle, containing the stages G1, S, and G2. The purpose of interphase is to prepare for mitosis, growing the cell and carrying out normal functions. In the G1 phase, the cell grows and carries out functions. In the S stage, DNA is synthesized or replicated. And the G2 phase is the final growth phase before mitosis. It's the massive buildup before it tears apart. M phase is where mitosis and cytokinesis occurs after interphase is completed. While mitosis is 10% of the cell's life, it is made up of 5 stages. Hey, would you look at that? This cell is in G2 phase, all ready for M phase. Let's check it out. First, prophase occurs. The chromatin begins to condense, and now you can see the chromosomes with their sister chromatids. Centrosomes are organelles located outside of the nuclear envelope and literally have no purpose. Until now! Mitotic spindles made out of microtubules begin to stem from the centrosomes as they move away from each other. Next is prometaphase. Creative name, I know, before metaphase. The nuclear envelope that once trapped and protected the chromosomes inside is now disappearing. This allows the microtubules to connect with the kinetochores. Remember that terminology? The microtubules that don't connect to kinetochore overlap with the opposite microtubules. Now after prometaphase is metaphase. The centrosomes are finally at the opposite poles. Woo! The chromosomes that are attached to the microtubules are now lined up in a single file line along what we call the metaphase plate. So low-key looking like the Earth. You can see the poles, the longitude lines, and even the equator. Anywho, after metaphase, bang! Did you miss that? Anaphase is one of the shortest phases of mitosis. Let's rewind the clip. The microtubules connected to the kinetochores shorten and pull the sister chromatids apart. Now, the chromatids have upgraded to individual chromosomes. Remember the microtubules that didn't connect to the kinetochores? They also have a job. Rather than shortening, they lengthen, pushing up against each other and ultimately elongating the cell. Finally, telophase and cytokinesis. During telophase, as you can see here, the nuclei around the chromosomes are beginning to form in both the daughter cells, returning to their uncondensed form. Where did the chromosomes go? Back to the chromatin. Now cytokinesis occurs. This is an animal cell, so we can see the actin filament ring around the middle of the two cells creating a cleavage furrow. See how it's tightening, and pop! It contracted so much that it split the cells, creating two daughter cells. Now, the process of cytokinesis is a little different in plant cells, so let's go over to the whiteboard to see what happens. Notice how the plant cell has gone through all of mitosis except for cytokinesis, since there are two nuclei in one whole cell. These little orange circles are membrane-bound vesicles. They begin to cluster in the middle and fuse their membranes together, creating what we call a cell plate. Now the cell plate will soon transform into a cell wall, which borders the entire cell, dividing the cell from 1 into 2. How cool is that? Mitosis is a crazy way to end this cell cycle, that's for sure. But with all the steps in interphase and mitosis, there are bound to be some mistakes, right? Just like how students double check their tests before turning it in for a grade, the cell cycle is composed of several stop-go checkpoints to ensure that there are no mutations. The 3 checkpoints on the cell cycle are the G1 checkpoint, the G2 checkpoint, and the M checkpoint. During the G1 checkpoint, the cell checks for the proper size of the cell, the appropriate growth factors in play, and to make sure the DNA isn't damaged right before synthesis occurs. Honestly, G1 checkpoint, I'm your biggest fan. You're so important to ensure that the rest of the cell cycle goes on with no bumps in the road. Whew. If the checkpoint gives the go, the cell will complete the whole cell cycle and split. But if there's a stop, the cell is sent to G0 phase. But don't worry, G0 isn't a bad place. It's a stagnant stage in which the cell doesn't go through mitosis. Some cells in G0 will never split again, carrying on their normal functions for the rest of their life. Eesh! Sounds like a 9-to-5 job. Other cells stay in G0 for a period of time, but return to G1 phase when it needs to split. Kind of like being put on the bench until you can be of use. Trust me, I know a bit or two about being a bench warmer. Softball wasn't my best sport. After a cell is able to pass the G1 checkpoint, they go through DNA synthesis in S phase. Then they are stopped at checkpoint G2. Here, the cell double checks that the DNA was replicated accurately and the DNA isn't damaged. If the checkpoint gives a go, the cell proceeds on to mitosis, the final step. But if they get a stop, the cell cycle is put on pause and the cell tries to correct its mistake. If the cell doesn't get the mutation fixed, the cell will undergo apoptosis. A dark time indeed. But sacrifices must be made, so no mutated cells are multiplied in the organism. After they pass G2, they go to mitosis, completing prophase, prometaphase, metaphase, then stop! The cell is held in its position in metaphase on the metaphase plate, looking like the Earth or something, as they have hit the M checkpoint. The cell ensures that the microtubules have appropriately connected to the kinetochore and the others are overlapping. If they receive a go, they continue to finish up mitosis, becoming 2 beautiful daughter cells. I'm so proud! But if they receive a stop, the cell will pause once again in the cell cycle and wait until the spindles finish attaching to the kinetochores. As soon as that's completed, they can pass. Bang! Right into anaphase. Man, I love that phase. Checkpoints are a good method the cells use so they can split properly through these crazy steps. The cell cycle is also regulated through internal and external regulations to ensure it continues smoothly and efficiently. Internal regulators consist of proteins called cyclins and enzymes called cyclin-dependent kinases. As the title practically says, the cyclin-dependent kinases rely on a certain concentration of cyclins to activate. The concentration of cyclins vary throughout the cell cycle, but as soon as the concentration reaches a sweet spot, the kinases fire and phosphorylate certain proteins, each to help regulate key events in the cell cycle. So, technically, if the cyclin-dependent kinases don't do their job, it's not really their fault. Stupid cyclins. External factors help regulate the cell cycle as well. Growth factors release hormones that set off the signal transduction pathway in order to stimulate the cyclin-dependent kinases. Contact inhibition depends on receptors on the cell's membrane to identify contact with the other cells, halting the G1 phase. Take a look at this jar of cells, for example. The receptors on the cell membrane detect contact, which means that the volume is full and there's not enough space to divide. But take a look at this example. The receptors on the top cells don't have anything in contact with them, so they know they're able to go through mitosis and know to stop when the volume is full. Even though the cells have these enhanced mechanisms that allow them to double check and regulate the cell cycle, there are incidences where mutations slip past. Cancer is the abnormal growth of cells in a certain area or through the body. While normal cells stop appropriately at checkpoints, the cancer cells neglect to follow, speeding through the red stop signs. Growth factors such as contact inhibition don't affect these cells, as they jam themselves together and divide indefinitely. The uncontrollable growth of cells often forms a tumor. Leukemia is a form of cancer that is located in the bloodstream. Yeah, the circulatory system that travels throughout an organism's body. Leukemia causes excessive growth in white blood cells from infected stem cells, and white blood cells are unable to fight illnesses as well as red blood cells or platelets. This often causes organs to not function properly. Leukemia is similar to all cancers, as there is an abnormal growth of cells, white blood cells, that causes organs to lose function and not operate as they should. One way to prevent this form of cancer is to refrain from smoking or being around excessive radiation. Cancer can be a devastating illness, as your own body has turned against you. Do what you can to avoid getting it, and pray for those who have it. Well, that's all we have today. The cell cycle is a fluid and efficient mechanism that allows you and I to function properly and live our lives. Thanks for watching, and I hope you pass your AP Biology test or whatever class you may be taking. Seriously, nobody watches this stuff for fun. This has been Dr. Schuliger. Hope you enjoyed. Have a great day and see you next time.